Hi guys, this is so that today we are starting from the urea cycle. So what I want to say you here, what you see is a overall reaction of the urea cycle. Overall reaction. That means you have to know it has several intermediate steps. And also that, here there has to be two water molecules and two ADP, and so here that PP1 or PPI means pyrophosphate, right? So that what you have to know here is the basic ingredient of the making urea. You need ammonia, carbon dioxide, and also at one stage aspartate also included. So you can research and find about the overall urea cycle. In some textbooks, right, they have taken out aspartate and they have started with the ammonia, right? So this is the important thing is this is an irreversible reaction. This is an irreversible reaction. That means it's happened only one way, right? Glutamate is a precursor of the ammonia, right? So oxidative deamination by glutamate dehydrogenase, aspartate nitrogen, transmination of oxaloacetate by AST. So you have to know about the meaning of the oxidative deamination. Oxidative deamination. So oxidative deamination is what? So oxidative deamination is a biochemical process by which amino group is NH2 group, is removed from an amino acid, producing keto acid and ammonia, NH3. So oxidative deamination is, so you need to understand these things when you are learning biochemistry, right? So oxidative deamination is, it's obviously biochemical process. So by which amino group is NH2, some is removed from amino acid. So then during the process you get a keto acid and the ammonia NH3, right? So now you have to know that the next term, right? Transmination. What is transmination? Right? Transmination is what? So we can discuss transmination related to urea cycle. So it's a process of uh, amino group of one amino acid is transferred to keto acid, forming new amino acid and new keto acid. Transmination, right? It is very important you to know these meanings, right? Please write it down and study those meanings: oxidative deamination and transmission of oxaloacetate by AST, right? Urea cycle. So urea cycle, that if you think about urea cycle, so you, different authors have explained in different ways. So here that five-step urea cycle. So place of reaction, mitochondria and cytosol in the liver, right? So regulation, you know the mitochondria and cytosol, what it is in cell, right, in the liver. Regulation of the urea cycle. So N-acetyl glutamate is an essential activator for carbamoyl phosphate synthesis 1. Carbamoyl phosphate synthesis 1. Right. So this one in the standard biochemistry textbook, so you will get more information. So hepatocyte is the liver cell. Hepatocyte is a liver cell. Right. Hepatocyte is the liver cell. Right. So now, any standard biochemistry textbook gives you more information about that, right? Now, coming back to the urea cycle again, right? So here, step one, formation of carbonyl phosphate. So location is mitochondria. Okay, please check it. Where mitochondria, there's a stroma inside the mitochondria, and also that there's, in A level we might have learned there's a mitochondria is a double membrane. So in double membrane, there's a reaction happen in between double membrane. Reaction happen in the inside the mitochondria. So, and also there's like a stroke particles, right? So these things we are not going to talk about that structure of the mitochondria. But anyway, it is better to study, have a little look about the structure of the mitochondria as well to recall our knowledge, right? So now here formation of the carbamoyl phosphate, location mitochondria, substrate, carbon dioxide, ammonia, and 2-ATP. Enzyme, carbamoyl phosphate synthetase, activator N-acetyl glutamate act as a positive allosteric activator of CPS1. Right. Product is a carbonyl phosphate. So now here the second part, formation of citrulline. Formation of the citrulline. Right. So I told you that you have to study about the mitochondria. So mitochondria, there's a matrix. So some people say it's a stroma. So matrix of the mitochondria. And also that, so if I draw about that one, right, because this is kind of a little bit above than the A levels. So it's a sausage shape. So this is the normal appearance of the mitochondria. So you get enzyme particles. And here there's a one name, here there's another name. So those things you can research and find so that I'm not going to discuss those things here. Right. So now here urea cycle, formation of citrulline and location mitochondria, substrate carbonyl phosphate and enzyme ornithine transcarbamylase. That is OTC, but don't worry, they are not going to ask what is OTC. Reaction, the carbamoyl group is transferred to ornithine, right? Product, citrulline and high energy phosphate transported to the cytosol, right? So then step 3, synthesis of argineosuccinate. So this is happening in the cytosol. Right, so that don't ask what is cytosol and the function of the cytosol. I told that those things because you know that when you're learning cytology for your A levels, so you might have learned about the structure of the mitochondria and also the structure of the chloroplast. Right, so those are basic things. So you can go through and understand those things if you need additional information. Right, if you need additional information. Step 3, synthesis of the argin argininosuccinate. So that if you want to pronounce this, you break this into parts and pronounce. Argini, Argini, argininosuccinate. Location cytosol, substrate, citrulline, aspartate. So enzyme, argininosuccinate synthetase. Reaction, carbide citrulline with aspartate to form argininosuccinate. Aspartate provides the second nitrogen which is incorporated into urea. So ATP, PP1, I explained what is PP1 and AMP, right? So product is argininosuccinate. Urea cycle again, location cytosol, substrate argininosuccinate. So enzyme, these are important. Enzyme. So what is the enzyme? Succinate lyase. Reaction argininosuccinate is cleaved to yield argin and fumarate. Fumarate can be hydrated to malate. So malate re-enter to the tricarboxylic acid cycle. So it's oxidized to oxaloacetate and can be used to gluconeogenesis. Gluconeogenesis can be converted to aspartate via transmination. AST product arginine fumarate. Right. Next step. Right. So now here cleavage of arginine to ornithine and urea. Location cytosol, substrate arginine, enzyme arginase 1. ARG1 primarily found in the liver. Reaction arginine is cleaved, cleaved to form ornithine, ornithine and urea. Other tissues can synthesize arginine, but only the liver can cleave arginine to urea. So this is important. Arginine to urea. Arginine to urea. So those steps, right? So now the important thing is, now it is we are done. Actually, this is a very simple explanation for the urea cycle. We can have a look about it again. Regulation of the urea synthesis. So N-acetyl glutamate, essential activator for carbamoyl phosphate synthetase 1, synthesized from acetyl coenzyme and glutamate by N-acetyl glutamate synthesis. So arginine is an activate. So after protein intake, amino acid level increase, then intrahepatic concentration of the N-acetyl glutamate increase, rate of the urea synthesis increase. So that put a star mark. This part is important. Fate of the urea. Urea is transported from the liver. Urea is transported from the liver, then kidney excreted in the urine. A portion of the urea, so go to blood and intestine, cleaved by bacterial urease, then carbon dioxide and ammonia. Some exceeded in the feces and some reabsorbed into the blood. In patients with the kidney failure, plasma urea levels are elevated, promote transfer of urea to gut. Bacterial urease convert urea into ammonia. Experience hyperammonemia. Right. So next one is a clinical condition. So it is hyperammonia. Hyperammonia. Hyper hyperammonemia. Right. That is how you pronounce. Right. Characterized by increased level of ammonia in the plasma, high morbidity and mortality. Normal range less than 50 micromole per liter. Highly associated with the compromised liver function. Disrupt the urea cycle caused by inborn genetic mutation. So you need to understand about the clinical condition hyperammonemia. 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 Right. Caused by inborn genetic mutations. Liver disease. Symptoms: tremors, slurring of speech, vomiting, cerebral edema, and blurring of vision. High concentration ammonia can cause coma and death. Right. So these are the, normal range of the, what is it? Right. Normal range of the, is it ammonia? Right. We'll see what is the normal range. Actually, that this is the normal range of the ammonia, right? Right. So that you need to know, I don't think that you have to remember these reference ranges, right? So normally it will be given. But since neonate, this is the amount, this is micromole per liter. So infant, 10 days to 2 years old. So 40 to 80 micromole. So you can see now here, adult, what happened? Ammonia level decreases as as the person age, right? So these things, right, better to have some idea, right? Hyperammonemia. So acquired it was difficult to say, hyperammonemia due to liver disease, viral hepatitis, hepatitis B, hepatotoxin, alcohol, cirrhosis, formation of collateral circulation around the liver. Portion blood is shunted directly into the systemic circulation and does not have access to the liver. No conversion of ammonia to urea, elevated level of ammonia. Right. So there's another one here you have to know that to some extent, right? Congenital hyperammonemia. 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 hyperammonemia, right? So even it's something funny, even if you see the pronunciation of the Indian doctors, so they pronounce in the different way. Hyperammonemia is the better way. That's how we pronounce hyperammonemia, right? So that you know that there are a few types you have to know. Arginase deficiency, right? Arginosuccinate lyase deficiency, arginosuccinate synthase deficiency, and the ornithine transcarboxylase deficiency, carbamyl phosphate synthesis 1 deficiency. So description also, it is better to know this table, it is important, right? So next condition is what? Next condition, azothemia, right? Azothemia is what? So azothemia, that some people misunderstood this condition, right? So azothemia, especially when you are learning biochemistry, so it is better you should know these basic terms, right? Now we'll see what is azothemia. Azothemia is, what is this? Elevated nitrogen waste. Sorry, I said it in a wrong way. Azothemia, right? So BUN, blood urea nitrogen and serum creatine levels. Reference range BUN, that means BUN is 1. So 8 to 12 milligram per deciliter, and serum creatine 7 to 1.4 milligram per deciliter. Increased level of BUN and serum creatine is high protein diet and also kidney failure, right? So that's it. Thank you. Now you can ask questions. The important thing is here when you're learning biochemistry, so you got to remember so much of terms, so many terms. Therefore, it is very important you to go through, and also that in end product and starting product, end product and starting product. So that is also important. For example, now here Urea cycle, so 5 steps, because I told different authors describe in different way. So even here, they have given overall equation. I told that you have to balance certain parts of the equation. So better you refer the textbook and rewrite the urea cycle equation, right? So 5 steps, so mitochondria and cytosol in the liver and regulation of the urea cycle. So N-acetyl glutamate is an essential activator for carbamyl phosphate synthase, right? So urea cycle, step 1, step 2, step 3, step 4, step 5. So product is ornithine and urea. And also here you have to know arginine to urea, right? So then here the location, cytosol and arginine. So I explained these things, right? Okay. Now thank you for joining, and we'll talk more about that later.